yesterday we studied about the breaking the triglycerol and the glycerol bond going this pathway and it goes to the five organs, five important parts. And now we are going to study about the rest, that is fatty acid. It is converted to three fatty acid and glycerol bond now. So now the fat, what happens to the fatty acid is our topic. So this fatty acid through circulation goes to the mitochondria. Mitochondria is having two cells, two layers, means outer layer and inner layer. And this outer layer, there is an enzyme, it's acyl transferase. It will give the gate pass for this fatty acid to enter the outer layer, inside the outer layer. And from there, it wants to go to the inner layer because beta oxidation happens in the inner layer. So there is another gatekeeper, his name is carnitine acyl transferase. It will give a pass and then it goes to the CAT2 and it will give the pass to enter to the beta oxidation. So in beta oxidation, what is the result is that the fatty acid will become three important things, that is NADH plus H plus, FADH2 and acetyl CoA. So NADH plus H plus is 3 ATP, FADH is 2 ATP and acetyl CoA in Krebs cycle will become 12 ATP. So that I will explain the next. But so total one fatty acid will give you 17 ATP. So how this acetyl CoA becomes 12 ATP is the next thing. And so this acetyl CoA is coming to enter the Krebs cycle, and Krebs cycle happens in mitochondria. Okay, all these things are happening in mitochondria. So acetyl CoA coming to the mitochondria, there is a pyruvate dehydrogenase which helps to open the door for entering into mitochondria. Krebs cycle, sorry. So what happens is that there is a presence of oxaloacetate which has 4 carbons plus the 2 carbons of acetyl CoA. CoA is a star written here because it is not a carbon, it's a driver who gives the way, who drives them all the way around this Krebs cycle. So this 4 plus 2 carbons will give you citrate which has 6 carbons. Okay, so now this citrate will become isocitrate, and from there is an enzyme named isocitrate dehydrogenase will remove one carbon and release one hydrogen. So what is present there? There is B3. B3 is NAD. It captures the hydrogen and becomes NADH plus H2, and it is stored inside the Krebs cycle. Maybe we can consider it as an inverter battery. It's saving energy for future. Okay, so one NADH plus H plus is saved there, and from there the next it goes to become alpha keto glutarate, alpha keto glutarate. Okay, and from there is an enzyme present, alpha ketoglutarate dehydrogenase will remove one carbon and releases H. So there is another NAD present there, B3. It will also capture the hydrogen and it becomes NAD plus NADH plus H plus, and it also stores the NADH inside, NADH plus H inside. And then this alpha ketoglutarate will be converted to succinyl CoA, and this succinyl CoA will be converted to succinate by a succinate CoA enzyme, a kind of enzyme, by releasing the CoA. It releases the CoA, and there is a product GDP plus inorganic phosphate will become GDP. GTP. GDP is guanosine diphosphate and GTP is guanosine triphosphate, and GTP is equal to 1 ATP. So that's also stored here. So we got 1 ATP extra, and from succinate then it is converted to fumarate by another enzyme, and what happens is that now no carbon is losing, but the hydrogen is captured by B2, FAD, and it becomes FADH2, and it is also stored in this. And after fumarate, it becomes malate, and from malate, it becomes again back to oxaloacetate. So the Krebs cycle starts with this mixing acetyl CoA with oxalate and also ends with oxaloacetate. Okay, so from becoming malate to oxaloacetate, there is a B3 NAD who captures this hydrogen and become NADH plus H plus. So that also is stored here. So when we calculate 1 ATPs plus NADH H plus is 3. So 3, 1, 4, 7, 9 and 12 ATP. This is how the Krebs cycle works.